Good morning, everyone. My name is Aliza Hutley, and today we will be discussing ion exchange chromatography. So, what exactly is chromatography? Well, it's an analytical technique that enables the separation, the identification, purification of the components of a mixture for qualitative and quantitative analysis. Some terminology that you want to be familiar with before we dive into the different types of chromatography is the mobile phase or carrier. And so this particular term means it's a solvent moving throughout the column, as you can see depicted in the pictures on our right. The next term that you would want to familiarize yourself with is the stationary phase, which is essentially the absorbent or the substance that stays fixed inside the column. So if you see the first depiction where it says column packed with a silica gel. In this column, you have your stationary phase. This is the phase that does not move, essentially. And then the next term is eluent. This is a fluid that enters the column. The eluate is the fluid that exits the column that is collected in the flask. And then the elution is the process of washing out a compound through a column using a suitable solvent. And then the analyte is essentially just the mixture whose individual components has to be separate and analyzed. And so the picture on the right shows a good depiction of a chromatography process. So this one here is a liquid chromatography process where they're separating the solution into different colors based off the colors. And so that's just chromatography in general, but there's many different types of chromatographies, including column chromatography, which was on the previous slide, ion exchange chromatography. You have gel permeation, affinity, paper, thin layer, gas. The list goes on. There's many different ways to separate substances. So that's what I like to think of chromatography. It's just a separation technique. So ion exchange chromatography is the focus of my presentation. I just wanted to get you guys familiar with chromatography in general, but now that we have an idea of what it is, let's focus on what exactly IEX is. That's the acronym for it. So this particular technique allows for the separation of ionizable molecules on the basis of difference in charge properties, so ions. And so it operates on the principles of electrostatic interactions between charged molecules and a solid stationary phase containing fixed charge groups. And so let me see if I can move this person out the way. Essentially, this picture on the right gives a depiction of ion exchange chromatography via columns, where you are using the columns to enter your solution or your sample mixture. And the elution buffer within the column is going to be either charged positively or negatively. And so we'll get more in depth on the next slide where we discuss that the stationary phase can be made up of a resin with either a positive charged group, which is known as cation exchange, or a negatively charged group, which is known as anion exchange. So that sample that we saw on the previous slide contains a mixture of charged molecules, which is loaded onto the column. And as the mobile phase that moves, it flows through the column, the molecules will interact with that charged group on the stationary phase, the one that remains still. So almost like you're washing the column out. And so depending on the charge of the resin that's within the column, the molecules will either attract or be repelled by the stationary phase, leading to the separation. So it depends on your resin, which type of ion exchange you're going to use. So we'll get more in depth within the type of exchange, which is following next, cation exchange. In cation exchange chromatography, a negatively charged cation exchange resin is used. So the resin that's within the column particularly is what's going to determine the difference in these charges. So the resin consists of small beads coated with negatively charged groups such as carboxylic or sulfonate groups. These negative charges on the resin attract and bind proteins with a net positive charge at the working pH. And here's a picture of the charges binding. And then here we have ion exchange, which is quite frankly the opposite, if you ask me, but a technique for separating molecules based on their net surface charge. It employs a positively charged ion exchange resin that is attracted to molecules with net negative surface charges. And so in this picture, we have a mix of amino acids. So imagine you throw a solution with the down a column or yeah, we're going to say column. And as that solution flows, you're going to have binding occur. So if you use the anion exchange, that binding of negatively charged amino acids to an immobilized cation surface will occur. And so essentially separation occurs because of binding. Depending on your solution, on the type of resin you use, it's gonna stick to the surface of the resin, and then the stuff that you either don't want or do want will flow right out. It depends on which type of chromatography you're performing. Alrighty, so industrial applications of ion exchange chromatography includes protein purification, pharmaceutical industry, environmental analysis, nucleic acid separation, and biotechnology. And on the right, you have a TCAN Freedom Evo picture because this is a piece of equipment that I myself use in the industry. I work for Eli Lilly for the summer. I'm interning there, and we do protein purification. And so the way we go about this is that I'm currently conducting resin screenings, and this particular instrument helps me build the plates that I need that will do the wash and elution steps and flow through plates for my resin. So yeah. So that's where I'm going to expand on. Like protein purification is one of the examples that I choose to talk about because I'm familiar with it. But because my research is private and it's not published yet, I had to pull an example from a paper where ion exchange chromatography is being utilized. It's one of the most common procedures for protein purification. So the example study that I pulled was their objective was to formulate a unique method that can be applied to purify any recombinant basic protein from E. coli. And so the materials included a cation exchange resin, an SP cephorose, and a selected buffer depending on the PI of the recombinant protein. And so using ion exchange chromatography, they're able to purify a protein that is like very difficult to obtain and mass produce. And so that's one of the benefits of using ion exchange chromatography and how it's used in industry. So this is also a slide that's dedicated to just showing you all the equipment that is used within industry. This is the red equipment is called an ACTA. Um, and then on your right, we have an HPLC system, and we're going to talk about what exactly that is. And so the ACTA is a versatile automated liquid chromatography system. So it's used for the fast purification of proteins and peptides and other biomolecules. So it's a wide range of things can be used for ACTA, but in my lab, we use ACTA for the purification of proteins. And so it's essentially utilizing the chromatography technique, allowing separation based off of the size, the shape, the charge. Is very many aspects of the protein of interest that it'll like take into account. And so the HPLC system, the one that was on the previous slide, is also an analytical chemistry technique used to separate compounds in a chemical mixture, but this one has a detector inside of it. So you can't see it on here. It's condensed, but it's like stacked little boxes on top of each other. And in this middle, you can see like they'll have these little lines where your liquid will flow through, and they change those lines and the sizes of it to conduct size exclusion. But it's essentially, if you open that HPLC system up, it'll have a detector that measures the analytes after elution from the column, and then it'll translate that data and it'll give you a diagram. It's going to be called a chromatogram. So both of these pieces of equipment gives us a decent amount of information needed to make analysis on our techniques and protein of interest. That concludes my presentation. These are some references that I've used to obtain this information for you all. If you have any more questions, feel free to contact me. Thank you.